The session on BCR microenvironment, I found it very interesting and very comprehensive. I thought the speakers gave an excellent overview of some of the key microenvironmental stimuli that are responsible for CLL development and progression. We had talks on toll-like receptor signaling and potential of novel IRAK4 inhibitors. We had a very interesting talk on monocyte macrophages that have been studied for a very long time, starting from the 2000 when nurse-like cells were identified as one of the major cell type that leads to CLL survival even in vitro, and nowadays to the relevance of macrophages in CLL in vivo mouse models and even in Richter syndrome development. And then we also heard about T cells and T cell dysfunction that accompanies CLL, and that is also driven by intrinsic features of the T cells that are dysregulated in the CLL microenvironment from an epigenetic standpoint. And then we also heard some very interesting talks on tumor-associated fibroblasts that are quite a novel topic. They're very well studied in lymphoma biology and very much responsible for lymphoma progression. We don't know much about the role of fibroblasts in CLL, and at the same time, they do seem to play a key role in CLL progression as well. And then we did hear interesting short presentations and brief communications highlighting the relevance also of macrophages and key factors that are involved, or signaling proteins that are involved in macrophages survival, and potentially in the crosstalk in between macrophages and CLL cells. And we also heard, of course, about the importance of integrin and CD49D, which is a key component of integrins, and that can facilitate homing and survival of CLL cells in the lymph node and the lymph node microenvironment. And it's a key prognostic factor. So I thought the session was incredibly interesting and very interactive, and it covered really a large number of topics that we have been hearing a bit about over the years, but there's still a lot to learn. There are still a lot of microenvironmental components that are incompletely understood, especially in the context of either refractory CLL or exactly in the context of novel therapies, and in particular, so also in the context of Richter syndrome. We're very much trying to understand determinants of transformation, determinants how tumor microenvironmental determinants change in transformed disease, and I think some of the aspects that were touched upon during this talk will also very much apply to Richter's transformation.